Namaskar dosto, welcome to channel Toxicology on Board. Today in this video we'll be discussing another DABT topic, that is Ricin and castor bean. So let's directly dive into the question. This question is taken from ABT decertification question paper only. The toxic glycoprotein in castor bean is. So if you look into ABT brochure, no, plant toxin are also an important topic in the case of syllabus, DABT syllabus. So one question with respect to castor bean is there. This question we'll try to read the various concepts with respect to ricin, the glycoprotein. Option A is tricinin, option B is alzerin, option C is brucin, option D is muscarine, and option E is ricin. The right answer will be in the green text, that is ricin. Ricin is the glycoprotein, which is the source is castor bean, that is Ricinus communis, and this is the one which is having adverse effects. So what are those adverse effects which is caused by this ricin that we'll discuss in this video? And meanwhile, you also pause the video, make note strychnine, which is coming from Nux vomica, alzerin, probably it might be used as a dye. Okay, brucin, which is coming, Ames, Bruce is there, from that it is coming, or muscarine, which is coming from atropine. So read all these things. Brucin, I am not knowing if I am telling the right. So go back and check which are the things, what are the sources, and what are those adverse effects. But this video will only discuss about the resin. Okay. So additionally, in the Clayson textbook, also read table 26.7, which they have listed out the selective plants and the who's having adverse effects on the kidney. So in that, castor bean is being listed, and the source is coming from this Ricinus communis. Here you can see the plant and all parts of the plant. So whenever you're reading about a plant, you have to read which part of the plant is responsible for causing the toxicity. So in this case of castor bean, all parts of the plant are responsible. They're containing this toxic glycoprotein, that is resin and ricinin, and they're having adverse effect on the kidney. So pause here. In addition, also read about other things are there, like you have water hemlock is there, mushrooms are there, impala, daphne, and autumn crocus. So pause and read which parts are responsible, what is the source, and what is the toxic which is released from this particular plant part. Okay, so let's restrict again, going back to resin only. Important points which are discussed under cassette and dose summary we'll read here. Resin is extremely potent toxin. It is isolated from the castor bean, that is Ricinus communis. Another source is same type of similar toxin can also be seen in the case of rosemary pea. Lectins are composed of two polypeptide chains connected by a disulfide bridge. How this alpha A chain and B chain, I will show you in next slide. One chain binds to the intestinal cell wall, that is A chain will bind, and because of which binding of A part of this particular, see here only it is A chain and this is the B chain. Uh, the A chain will bind to the cell wall, and as a result it will open it, which will allow the other chain, that is B chain, to enter the cytoplasm, where the ribosomal protein synthesis taking place is inhibited by fragmentation of these ribosomes. So basic mechanism is resin will have two chains, A chain and B chain. A chain will go and bind to the cell membrane, this phospholipid biomembrane, whatever it is, and it will enable the another chain, that is B chain, to enter into the cytoplasm. In the cytoplasm, where this particular ribosomal protein synthesis is taking place, that fragmentation of this particular ribosomes takes place, because of which protein synthesis is inhibited. A single molecule of the toxin can kill a cell. So these are the important points which are discussed under cassette. So go back and read in Clayson textbook. And another historically one incident that came across with respect to ricin is that the tiny platinum pellet killed BBC broadcaster. This is very famous news. Georgi Markov. This is the particular journalist. Journalist who is from Bulgarian defector, measuring just 1.7 millimeter. So in this platinum ball, no, resin was loaded, and what they have done, this particular person, no, he was standing in a bus stand. The person who wanted to kill him, he loaded that particular pellet, platinum pellet containing the resin toxin, inside this particular umbrella. So whenever he opened that, he pressed the button in the umbrella, it went and hit the thigh of this particular thigh muscle, intramuscularly thigh muscle of this particular journalist, and immediately the resin started spreading inside the body. it got once it was administered through intramuscular route, it entered into different spread into the different parts of our body, and immediately it caused adverse effects and death. So it is was filled with deadly poison, that is resin. An assassin injected into his leg using modified umbrella. This is the umbrella. It is still there in the museum. You can go back and read about this news. While Markov waited for a bus in the London, that is, this incident took place in 1978. So pause here, go back and read about this particular historical incident. Okay, so which of the following is true regarding the poisonous toxin in the castor bean? So you have to remember castor bean, common name, scientific name is Ricinus communis. The toxic glycoprotein is ricin. So with respect to ricin, what are the important statements, important right statements we'll see. The active principle is lectin. It consists of two polypeptide chains connected by disulfide bridge. This is the thing that we are discussing. This is A chain and B chain. One chain is essential for binding to intestinal cell membrane. I told you, no, and this binding leads to the entry of the other chain into the cell. That is, B chain will enter into the cell. A potent toxic in the intestinal cell wall. In the intestine cell walls will be there. No, this particular resin will go and cause the cell death of this particular cells, lining cells. So the right answer will be the green text. All the statements with respect to resin are true. Regarding the plant toxin resin, early manifestation of toxicity including nausea, vomiting and diarrhea upon acute exposure. So you have nausea, vomiting as well as diarrhea. Resin mechanism of toxicity is inactivation of ribosomal structure, as I told you, impairment of this protein synthesis, thereby blocking protein synthesis. Resin is composed of two peptide chains linked by disulfide bond. So if you read the important notes with respect to textbook, various years, see, earlier questions were from 2000. Now even 2004 also they have repeatedly 2 to 3, 4 questions have come across which were asked about resin in from the year 2000 to 2010 in the recertification examination papers. Okay, so let's see what are apart from ABT. Will in this video we'll discuss some questions from AACT also, that is American Academy of Clinical Sciences question of the day. Assuming that the question paper is set by the Americans, so there is a possibility of same kind of topics, important topics will be discussed under American Academy also. So from the question of the day, so first question is, resin is one of a number of toxin of plants and bacterial origin collectively referred to as ribosome inactive proteins, RIP. So under RIP, that is ribosome inactive proteins, apart from resin, what are there, that we will read now. Okay, so any questions with respect to other type of plant toxin also, if you get in exam, you will be able to answer it. Okay, they're so named for their ability to depurinate the alpha sericin resin loop SRL of a large rRNA and inhibit protein synthesis. Okay, they're going to inhibit the protein synthesis. What are the other members of this group of toxins that we'll see? So ribosomal inactive protein toxins. So first one is the plant toxin pokeweed. Pokeweed, different types of weed type of options you will be getting, ragweed, pokeweed. So you read, make a notes. If you make notes, it will be very easy at the time of revision of exam. So pokeweed antiviral protein PAP from Phytoleuca americana. The source, the plant is Phytoleuca americana. Then trichosanthin TCS from Trichosanthus kirilovi. Abrin, Abrus picatorius from Abrus picatorius. So many questions on Abrin also you'll get in exam. Then saporin from Saponaria officinalis, Gelonin from Gelonium multiflorum, Shiga toxins found in the bacterial species Shigella dysenteria and Shiga toxigenic Escherichia coli. So these are the various plant sources as well as one example with respect to bacteria. So all these toxins are ribosomal inactivating protein toxins. Okay. Next question we'll see. What is the antidote for resin poisoning? So whenever when you're discussing about any toxin, there will be, there not always, there might be some antitoxin also. With respect to resin, there is no antitoxin. So that concept we'll read in this question. What is the antidote for resin poisoning and what is the role of hemodialysis in the treatment of resin casualties? So once you have a poisoning exposure, whenever that particular patient is brought to the emergency department, there are different steps will be there. Airway, breathing, ABC will be there. And If you have an antidote, if you identify that poison exposure, then you can give the antidote. And there is another method also in that, one is hemodialysis. So in order to further uh, inhibit the absorption or remove that particular toxin from the blood, you will be doing hemodialysis. So here the question is with respect to that also, what is the role of hemodialysis in the treatment of resin casualties? The answer is there is no antidote against resin toxicity. As of now, currently there is no toxin antidote for this toxin resin. Resin is not amenable to remove by hemodialysis, so hemodialysis step also cannot be done. So how we are going to do the treat the patient with resin exposure? Only preventive, that is only management. That is treatment for resin related toxicity essentially involves supportive care. So you can only treat the symptoms, but you cannot detoxify that particular toxin. Okay. So pause here, read. So one question was with respect to ribosomal inactivating protein, and one concept was with respect to treatment of resin poisoning. Okay. Next, again we'll discuss. So in American American Academy of Clinical Sciences also they have discussed this particular incident. So in detail we'll discuss about this particular incident. So what has happened? So briefly in the first slide we have discussed that umbrella, how the it was loaded in the platinum pellet. So with respect that concept only we'll discuss here. What is the only reported case of human poisoning by ricin administered by intramuscular route, and what was the resulting histopathology reported in that case? So since it was administered, it was given in from the umbrella into the muscle intramuscular route, no. So after, what was the histopathology also we'll discuss here. Only reported case of human poisoning by ricin administered via intramuscular route involved Bulgarian dissident writer Georgi Markov, who was murdered by ricin injection in the case in the 1978, that is in the London. The author of the so from the pellet, I told you, no, platinum pellet, which was introduced into his thigh muscle. In the thigh muscles, they have given resin was distributed around his body, and many tissues were affected. Damage included presence of several local lymphoid necrosis in the inguinal lymph nodes, which drained the injection site from the lymph nodes, liver necrosis, diffuse inflammation of the kidney and the spleen. Hemorrhagic necrosis of the small intestine was also noted, as well as pancreas and testes. See how vastly the resin is spreading to various organs in the body. If it was given through oral route, it could have been different. Since it is given through intramuscular route, through lymph nodes, inguinal lymph nodes, so it has spread to kidney, to the spleen, to the liver, to the pancreas, to the testes, to the small intestine. Okay, myocardium, including heart. Myocardium and conductive tissues were found to have petechial hemorrhages, which also featured in the case of adrenal, gut wall, as well as lymph nodes. The key feature of a mild pulmonary edema, that is in the lung also, observed, was thought to have been secondary to the cardiac failure. Primary is the heart failure. The key feature of toxicity via the intramuscular or intravascular route is the fairly rapid distribution of the toxin. That's why route of administration is very, very important. Since the poisoning was through intramuscular route, the amount of damage it caused to this particular person was more. Okay, distribution of toxin around the body to most organs, making therapeutic treatment a very much challenging to the uh, clinical toxicologist. Okay, so pause here, read about this one, how this incident taking place, so that you can connect and remember. I'm not saying you'll get it in the exam, this particular incident, but try to understand route of administration is important, how the source, everything, right? Then something we'll discuss about how ricin used as a bioweapon during the World War, whether it was used or not. That concept will be reading in this particular two questions, sir. So first question is, during the World War II, the British military developed a munition termed the W bomb. So, they during World War II, no, a lot of bioweapons were prepared by the scientists. So, the one which was prepared by the British military is W bomb, and the major component in this W bomb is containing so-called compound called W, which was compound W. The compound W is nothing but it was containing resin. This so-called W1 was never used. That's why we have historical only one particular incident where it was used. Okay. Next question we'll see. Recent intelligence reports suggested that Al Qaeda Yemeni branch may be seeking castor beans for making resin. So a lot of security agencies will be there doing spy. So they have found that this particular terrorist group, they were in search of this resin in order to prepare it as a bioweapon. American counterterrorism officials are increasingly concerned that the most dangerous regional arm of Al Qaeda is trying to produce lethal poison resin to be packed around small explosive for attacks against the United States. Okay, so what is the resin content of this castor beans? So you know, the concentration is also very important when it comes to plant. How much percentage of that particular component, because in the case of plant, various mixture of glycoproteins, glycolides, alkaloids will be there. So what is the component which is in higher concentration, which is responsible for causing the toxicity, is important. So the question is with respect to what concentration, what percentage of this particular component is there in the case of castor beans. So the reports regarding the resin content of castor beans vary, but it is probably is in the range of 1 to 5 percentage. So whenever this concentration is up to 1 to 5%, so you can see how these toxins are used as a bioweapon also. Now, one question with respect to the mechanism, how the resin is causing impairing the protein synthesis. With this diagram we will be discussing. So the question is, describe the mechanism of action of resin. Very simple step. 4-5 steps, 3-4 steps are there. Try to remember. Resin is a protein derived from the castor bean plant. Consists of two chains. That is A chain and B chain. They are linked by disulfide bond. That's the ring. Next, resin B chain is responsible for facilitating the entry into the cell via endocytosis. Here you can see endocytosis will be there. That particular chemical agent resin will come in contact with the cell membrane. Then endocytosis will be taking place, then but that particular component will go inside the cell. Okay, resin A chain is then transported to the cytosol where it will bind and irreversibly inactivates, not reversible, irreversibly inactivate ribosomal 28S of ribosomal RNA. Okay, so thereby it is impairing the protein synthesis. Here it enters, it will go, it will bind to this particular membrane, endoplasmic reticulum, and it is going to impair the protein synthesis. The inactivation is due to removal of adenine residue and effectively halts the protein synthesis. Adenine, you have adenine AGPU, that is adenine guanine, which are purine and pyrimidine. Pyrimidines will be there, no? So it is removing adenine. Okay, thereby it is causing impairment of this. It is halting the protein synthesis. So hope from this you can pause here, read about how it is causing this particular mechanism. Okay. So from this video, hope you were able to learn about resin, the source castor bean, Ricinus communis, how it is affecting the various organs. Okay, we have seen one historical case study by intramuscular exposure. We have seen the mechanism also. We have understood the resin structure, like A chain and B chain, how it is impairing the protein synthesis, and we have also seen uh, how routes of administration are also important in order to cause the toxicity, and we have also seen the use of these toxins for preparation of the bioweapons. Thank you so much. Be curious. For any questions you can always write a mail to arya.priyanka18@gmail.com. All the best for your exam.